competitive. So coming to the competitive inhibitor, so competitive, the name itself says it just the competes, it just the competition between one substance to the another. So in this competitive, the competition will occur in both the substrate and inhibitor, since the substrate and inhibitor competitive inhibitor, the chemical that resembles the enzyme's normal substrate and compete with it for the active site. So normally active site is present in the enzyme. So active site is nothing but it is a part of the enzyme in which the substrate will bind in that active site and it forms the enzyme substrate complex and the products are released. So competitive inhibitor is the one which resembles like the normal substrate. So in substrate place the inhibitors will bind and inhibit the chemical reaction activity. So chemicals that resembles the substrate and compete it for the active site. So normally substrate will bind in that active site, but in this competitive inhibitor, the inhibitor will bind in that active site. So enzyme plus inhibitor does not form the enzyme inhibitor complex and hence no product has been released. So any compound that bears the structural resemblance particularly to a normal substrate of an enzyme and that competes with that substrate for binding at the active site of the enzyme. So the inhibitor which is not acted by the enzyme and thus prevent the substrate from approaching the active site. So hence the substrate resemble inhibitor will bind in that active site. So there are no more substrate binding takes place in that. So it causes the competitive inhibitor. So you could see that the diagrammatic representation where enzyme is marked. So active site is marked, it is a part of the enzyme and in that active site the substrate will bind. So you could see the substrate resembles inhibitor in the orange color. So that is the inhibitor. When that inhibitor is bind to the enzyme, no more substrate will bind and which causes no more product releases. So you could see the part B, inhibition. So enzyme is bind with the inhibitor and inhibitor competes with that substrate, hence no product has been released. So for example, so succinate. So succinate, which is a chemical compound which occurs in the Krebs cycle or tricarboxylic acid cycle or citric acid cycle. So that succinate which has to be converted into fumarate for occurring the chemical reaction. So in this malonate comes with the inhibitor activity. So malonate which resembles the structure of the succinate, hence in succinate place malonate will go and bind. So hence malonate is an inhibitor, there were no more reaction in the Krebs cycle. So the enzyme of Krebs cycle succinate dehydrogenase catalyzes the reaction of converting succinate into fumarate. The succinate converted into fumarate with the help of succinate dehydrogenase. So in this competitive inhibitor, malonate compound acts as the inhibitor because it has the similar structure to the succinate. So inhibitors bind to the active site of the succinate and inhibits the reaction. So next coming to the non-competitive inhibitor. So competitive we have understood that it competes with the substrate in the active site. So coming to the non-competitive inhibitor, it does not, the inhibitor does not bind to the substrate but it just bind to any of the site of the enzyme. So any of the part of the enzyme other than the active site. So coming to the definition, a non-competitive inhibitor which attaches certain allosteric site which is any site of the enzyme that is not the active site, because the enzyme has several parts. none other than active site. So apart from the active site, these non-competitive inhibitor binds with other parts of the enzyme. So it do not enter the active site but binds to the another part of the enzyme, so that the enzyme shape will be changed, and alternatively active site place can also be changed, and it is very difficult for the substrate to identify the active site of the enzyme. Okay, so this results in the shift in the three-dimensional structure that alters the shape of the enzyme and alters the shape of the active site, so that the substrate will no longer fit into the active site properly, because that inhibitor will bind into the any part of the enzyme, so that the enzyme shape also will be changed and the active site shape also change. So there may be no substrate attached to that active site or enzyme, and hence no product will be occur there.